Smooth muscle is also called visceral muscle and specialized for slow contraction and controlled by a number of involuntary inputs. Each thin, elongated, non-striated fiber of smooth muscle is enclosed by a fine network of reticular fibers, forming a very fine endomysium, which transfers the force generated by the contraction of each muscle fiber into some combined action such as peristalsis of the intestine. So the gut wall is a good location to show you smooth muscle. But you're going to see a lot of sections through smooth muscle when you study each body organ. So it's important you know its morphology when cut longitudinally or transversely, or even in oblique profiles. That will require continual viewing and a bit of experience. But here, I'll just explain the basic histological features you are likely to encounter. This is a section of the colon. Lining the scalloped lumen is a purplish stained mucosa, which consists of simple columnar epithelium over a connective tissue bed filled with tubular glands. This mucosa rests on a layer of loose to moderately dense connective tissue, the submucosa. And the next layer out, or more external layer, consists of a band of circularly arranged smooth muscle with the cells cut in the plane of section. The outermost layer appears as 3 larger bumps called tinea coli. These are longitudinally running bands of smooth muscle cut in cross-section. I'm now going to show you this region of smooth muscle cut longitudinally here and then transversely here. Smooth muscle cells, also called fibers, can vary in length. They're smallest in small blood vessels, about 20 micrometers long, and they're longest in the pregnant uterus, up to about 500 micrometers long. This is a relaxed sheet of smooth muscle. The single and elongated nucleus of each muscle cell is located in the center of the cell. The cytoplasm, or sarcoplasm, tapers at both ends, and the cells pack closely together, all orientated in the same direction. Note that the smooth muscle cell is not striated. The contractile filaments attach to dense bodies within the cytoplasm and on the plasmalemma, or the cell membrane, actually the sarcolemma. The dense bodies really are similar to the Z-disc in striated muscle. These cells are joined by numerous gap junctions, which allow the wave of contraction to pass along the muscle layers or muscle sheets. Each cell is enclosed by a very thin basal lamina, we call it the external lamina, and fine reticular fibers, or an endomysium. I'm showing you this image for 2 very important reasons. First, often you will see the nuclei in a corkscrew appearance, which occurs when the muscle fiber contracts. And this makes it a bit easier sometimes to distinguish a smooth muscle cell from a nearby fibroblast. And secondly, notice the difference between smooth muscle fibers here and larger collagen fibers up here. Then look at this next image. A longitudinal section through smooth muscle is much more cellular than you see in this image of a tendon, with the occasional fibrocyte wedged between collagen bundles. You can tell the lumen of this small artery is partially constricted because the nuclei of the smooth muscle cells in the artery wall have a corkscrew appearance. It's not really hard to imagine that this circular sheet of smooth muscle making up the bulk of the wall of this small artery can regulate the diameter of the vessel lumen and therefore the amount of blood it will pass to the tissue that it supplies. Now, in this image of the muscle in the colon sectioned transversely, you only see the nuclei when the muscle fibre is sectioned through the nucleus, or the widest part of the fibre. Other profiles are through the tapered ends of the fibres. The much paler stained external lamina and endomysium between the cells is fairly obvious.